Hello friends, a very good afternoon to you. Today, I am myself Dr. Pooja Johari and I will be discussing the biochemistry portion of Grand Test 184. The biochemistry questions are from 221 to 237. From 221 to 237. Now, let's see question number 1. Question number 1 is defective proteins are degraded by after attaching covalently to. Now see, whenever we talk about the defective proteins, once their function is over, once they are of no use, they actually bind with ubiquitin. The process is known as ubiquitination, forming a proteasome type of structure, and this process requires energy. So this defective protein will be acted upon by this ubiquitin. Basically what happens is there is an interaction between glycine residue of ubiquitin and lysine residue of the protein which is defective. So these two react and the degradation occurs. So let's see the option. Options are defective proteins are degraded after attaching covalently to. So the answer here is ubiquitin, and the entire process is known as ubiquitination. This is actually responsible for the proteolysis of the protein. Again, one very important concept about this is mediated with the help of ATP, and excessive proteolysis occurs. What are the other options? Clatharin, pepsin and laminin. Now clatharin is obviously related with LDL, pepsin is an enzyme, and laminin is again, it is the incorporation of the cell membrane. So the answer to this question is ubiquitin. Next, question number 2, function of exonuclease. Now see, exonuclease, as it is clear from the word itself. Exo means outside, and nuclease is the enzyme which is digesting the nucleic acids. There are two types of exonucleases enzymes. One enzyme is 3' to 5' exonuclease. Another is 5' to 3' exonuclease. Now see, if we have to differentiate between 3' to 5' and 5' to 3', you can see the direction of the strand cutting. If it is from 3' to 5', that means it is going to degrade the nucleases here. And if it is from 5' to 3', it is here. An exonuclease is a very, very important example of proofreading. Basically, see what happens is, let's see the question first. Question is function of exonuclease. It is proofreading. जैसे for example हम लोग exam में, when we appear in the exam, before submitting the answer sheet to the examiner, we go through it once and twice so that if there can be any chances of correction, we can make it. Same is the case with this proofreading activity. This is a characteristic feature of proofreading. Is a characteristic feature of DNA polymerases, which is actually a fidelity enzyme of replication. Once the DNA strand is already made, this DNA polymerase is going to move around the entire strand just to check mismatch base pairing. Mismatch base pairing is, say for example, adenine के साथ thymine is binding, instead of thymine this guanine will come. So obviously if there is mismatch base pairing, there is formation of thymine-thymine dimers. It is going to change the reading frame of DNA. Once the reading frame of DNA is changed, automatically there will be a change in RNA, or we will call it as a mutated DNA. So proofreading and editing is one of the very important processes of DNA polymerases 2, 1 and 3. 3 is basically involved in the processivity, or it is involved in the polymerization, but if we see the function of 2, it is actually related to repair mechanism, and 1. So there are simple other, you can say, processes by which this DNA repair mechanism occurs. First of all, we have to check whether there is mismatch base pairing or not, whether there is formation of thymine-thymine dimers is not. Then this DNA polymerase undergoes a process of DNA repair mechanism, which can be methyl directed strand cutting, methyl directed strand cutting, oligomer replacement, by chemotherapy or by oligomers. So the answer to this question is proofreading. Polymerization is simple. Polymerization means DNA polymerase is catalyzing the replication of DNA. Chain elongation is elongation of chain, it is clear from the word, and termination is once the process comes to an end. So exonuclease has proofreading and editing capacity. Then question number 223, for fitter, the shortest needle in pens should be. Now what do you mean by this fitter? This stands for, the full form of this fitter is Forum for Injection Technique and Therapy Expert and Recommendation. These are actually the new insulin delivery recommendation. So obviously the principle behind this is that the pen should be 4 mm so that there is complete release of insulin only. We have to rotate the injection sites, and do not reuse the needles. So in this fitter technique we use the injections which have 4 mm and the syringes which are 6 mm in measurement. So the answer to this is A, which is 4. The next question, OTC deficiency causes. Now see, this OTC actually stands for ornithine transcarbamylase. Now see, some things are clear from the word itself. Trans means transfer of carbamoyl group to ornithine. What is formed when carbamyl group is transported to ornithine? It forms citrulline. So this is what? This is nothing but the second mitochondrial enzyme of urea cycle. Now this urea cycle is very, very important. There every year a question is asked either on the enzyme deficiency or the site of urea cycle. Now see, urea cycle occurs in the liver. It occurs in 5 steps. First and the second step occur in the mitochondria, and third, fourth and fifth occur in the cytosol. So what happens is, in urea cycle, the substrates are carbon dioxide in the presence of in the form of bicarbonate ions. It combines with ammonia to form carbamyl phosphate. The name of the enzyme here is CPS1, which is present in the mitochondria, and 2 ATP are going to form 2 ADP. And a very interesting feature about this is N-acetyl glutamate is a allosteric positive modulator of this enzyme. Now once this carbamyl phosphate is produced, this will combine with ornithine to form citrulline. This citrulline will combine with aspartate to form arginosuccinate. This arginosuccinate will now form arginine, which will be converted into ornithine, giving out urea. Now let's look at the enzymes. Here the enzyme is ornithine transcarbamylase. Here the enzyme is arginosuccinate synthetase. So synthetase, so obviously ATP is required here. Here it is arginosuccinase, and here it is arginase. When this arginosuccinate is converted into arginine, fumaric acid is given out. So this fumaric acid is an intermediate of TCA cycle. So here we can see urea cycle indirectly is connected to the TCA cycle also. Now here see what happens is, deficiency of CPS1 will lead to hyperammonemia type 1. Deficiency of CPS1. Deficiency of CPS2 will lead to hyperammonemia type 2, because the deficient enzyme is ornithine transcarbamylase. And a very interesting fact about this enzyme is absent in the brain. That is why urea cycle is not operative in the brain, one thing. And then this arginase enzyme is absent in the kidney. That is why urea cycle is not operative in the kidney also. So overall we can see first and the second step, they occur in the mitochondria, and third, fourth and fifth occur in the cytosol. Similarly, deficiency of arginosuccinate synthetase will lead to citrullinemia. Deficiency of arginosuccinase will lead to argininemia, and obviously deficiency of this will lead to hyperornithinemia. Now let's look at the question. Question says OTC deficiency causes. The OTC deficiency will cause hyperammonemia type 2. Type 1 is caused by CPS1. Argininemia is caused by arginase, and citrullinemia is caused by arginosuccinate synthase. So the answer to this question is hyperammonemia type 2. Then question number 225. Question number 225 says, role of ubiquitin in cell. Just now we have discussed ubiquitination is the process which is actually degrading this defective protein. There actually formation of proteasome type of complex is there which requires ATP. Options are energy dependent protein folding. No, it is not folding of proteins. Energy independent, since it is not related with the folding of proteins, it is simple degradation of proteins. And it is energy dependent. Just now I have explained this to you. It has to be dependent on ATP. And ubiquitation is actually a process in which there is a formation of peptide bond between what? Between the glycine residue of ubiquitin and lysine residue of the target protein, which is defective, which has to undergo proteolysis. So the complex is proteasome and the process is known as proteolysis mediated by ubiquitin. So the answer to this question is energy dependent proteolysis or protein degradation. Next question is, Rothra's test is utilized for the detection of. This is a very simple question and very important question from examination point of view. Rothra's test, this question should be actually learned by you by default only. Whenever the concept of this Rothra's test comes into your mind, immediately it is ketone bodies. Now see what happens is, Rothra's test is a test for the detection of ketone bodies in urine. Now what are these ketone bodies? These ketone bodies are acetone, acetoacetate and beta hydroxy butyrate. Out of these three ketone bodies, acetoacetate is considered to be the primary one. If it undergoes dehydrogenation, it forms beta hydroxybutyrate. If it gives out a molecule of carbon dioxide, it forms acetone. So actually it is the acetoacetate only which is reflected. And the process of formation of ketone bodies is ketogenesis. The normal concentration of ketone bodies is 1 milligram per dl. But ketogenesis is increased in starvation and diabetes mellitus. These are the two reactions in which this ketogenesis increases. The reason behind this is same, whether it is starved state or whether it is diabetes mellitus, there is increased breakdown of the fats. When fatty acids break, they form acetyl coenzyme A, and this acetyl coenzyme A, which is the precursor of ketone bodies, will be converted to form these. So Rothra's test is given by acetoacetate. Another important aspect is कि only acetoacetate will give a positive Rothra's test. Means beta hydroxybutyrate will not give Rothra's test. This is again a very important question which is asked in the exam. So Rothra's test is the test given by acetoacetate, which is a ketone body. Now let's look at the other options also so that we can know what are the others. Glucose, proteins and urea. Now glucose के लिए again there are so many tests for glucose. If we take the example of glucose only, since it is a reducing sugar, it will give Benedict's test positive. It will give picric acid test positive. This is simple qualitative. If we have to quantify glucose, for quantification of glucose, we require what? We require glucose oxidase peroxidase test. Similarly, for proteins, biuret test is carried out. Biuret test and ninhydrin test are the confirmatory tests for every protein, and there are certain other tests which are given by specific amino acids. For example, xanthoproteic test. Xanthoproteic test is given only by aromatic amino acids. Hopkins-Colles test is specific for indole ring of tryptophan. Millen's test is specific for tyrosine. Lead acetate test is specific for cysteine, and Sakaguchi test is specific for the guanido group of arginine. So these are for specific amino acids, but overall if we see, whenever we have to quantify also proteins in vaccines, for example, we carry out this biuret test. Then next, so here the answer is Rothra's test is utilized for the detection of ketone bodies, and Rothra's test is given only and only by acetoacetate, which is the primary ketone body produced. It is not given by beta hydroxybutyrate. And if we talk about acetone, acetone is volatile, it will go off. So it is given by only this. The next question, 227, selenium is essential for. Now see, again this is a very, you can say, simple question. Whenever the concept of the selenium comes, the major function which is catalyzed by selenium is 227. It is the trace mineral which functions as a antioxidant. And this is the major component of the enzyme glutathione peroxidase. This is again a very important question. Now what is this glutathione? This glutathione is a tripeptide formed of glycine, cysteine and glutamic acid. The function of glutathione peroxidase is mainly in RBCs. Basically what happens is, glutathione exists in two forms, reduced form and oxidized form. To make glutathione in reduced form, it requires NADPH. This NADPH which is coming from HMP shunt. Now when this glutathione is in reduced form, it can act on hydrogen peroxide. This is the major role of an antioxidant, that it is actually preventing the release of free radicals, and selenium is required. And now what happens is, if suppose this NADPH is absent, this glutathione won't be in its reduced form. So this hydrogen peroxide is going to release peroxide free radicals. And these free radicals have a very, peculiar feature, that they are in search of an electron. Wherever they find an electron, they combine with it, or they start eating the cell membrane. and that is what exactly happens. Deficiency of NADPH या deficiency of glucose 6 phosphate dehydrogenase leads to excessive hemolysis or hemolytic anemia. So here the function of an antioxidant is that it is preventing the formation of free radicals, and here the question is what? Glutathione peroxidase, reductase, synthetase, hydroxylase. So obviously it is glutathione peroxidase. The next question, what is the name given to the relaxation process due to an interaction between an excited nucleus and the magnetic fields caused by nuclei moving around in the sample? So it is nothing but the relaxation of the spin and the lattice. So here the answer is spin lattice relaxation. Lattice relaxation. It is defined as the mechanism by which the component of the magnetization vector along the direction of the static magnetic field reaches an equilibrium with its surroundings by obviously a resonance. There is an equilibrium by resonance characterized by this spin lattice relaxation. So the answer to this question is A, spin lattice relaxation. The next question 229, all of the following are antioxidant enzymes except. Now antioxidants, again it is clear from the word itself, which are against oxidation, or you can say which won't allow. In a simple language, if we say antioxidants are those which won't allow the proliferation of the free radicals, which will stop the proliferation of these free radicals. This is the function of an antioxidant. There are so many antioxidants which are present. If we talk about vitamins, vitamin E is a very important antioxidant. Vitamin A is an antioxidant. If we talk about the level of the enzymes, superoxide dismutase is a very good example of this. Catalase is an example of this. Similarly, ceruloplasmin is an example of this. These are what? These are functioning as antioxidant, means they are actually, see what happens is, if we see the mechanism of free radicals. Free radicals undergoes 3 steps: chain initiation, chain propagation, and chain termination. The function of these antioxidants is to terminate the chain or preventing the oxidation of whether it is oxide radical, whether it is peroxide radical. So let's look at the options. Catalase, yes, it is an antioxidant. Glutathione peroxidase, just now I showed you the example of this. It actually prevents the formation of peroxide free radicals. Same is the case with catalase. Ceruloplasmin to some extent is also working as an antioxidant. So the odd one out is xanthine oxidase. So xanthine oxidase is not a antioxidant, and xanthine oxidase is the enzyme which is required actually in the conversion of hypoxanthine xanthine and then to uric acid. So it is not an antioxidant. Then question number 230. In what form does the product of glycolysis enters TCA cycle? Now see, this is again a very interesting question, I must say. What happens here is glycolysis immediate pathway of oxidation of one molecule of glucose in the presence of oxygen or in the absence of oxygen. It always forms, glucose has 6 carbon atoms, it always forms 2 molecules of pyruvic acid, which is produced in the cytoplasm. Now this pyruvic acid, when it has to undergo oxidative decarboxylation, this is simple dehydrogenation in the presence of PDH enzyme, which is an example of multi-enzyme complex. Here oxidative decarboxylation means pyruvic acid has 3 carbon atoms. There is a removal of this carbon dioxide. We are left with what? CH3, C double bond O, SCOA. This is nothing but this is acetyl coenzyme A. This reaction is taking place in the mitochondria. Why? Because once this acetyl coenzyme A is produced, it is having 2 carbon atoms, it will undergo degradation by this TCA cycle, where it is going to give out its 2 carbon atoms in the form of carbon dioxide here, where ICD enzyme is present, and here where alpha ketoglutarate dehydrogenase is present. So the answer to this question is, in what form the product of glycolysis, it has to be acetyl coenzyme A, because it is the first substrate, you can say, of TCA cycle. It will now combine with oxaloacetic acid to form first stable product, that is citric acid. So answer is acetyl coenzyme A. Next question, how do we define oxidation? Very simple, oxidation is defined as the loss of electrons. Actually, there are 2 parallel processes, one is oxidation, another is reduction. Reduction is gain of electrons. And this is a typical feature of a redox reaction, where oxidation and reduction takes place simultaneously. If a substrate is reduced, if a substrate is giving out electrons, it will be converted into this. So this is being reduced because it is accepting this, and this is being oxidized. So oxidation is defined as the process of loss of electrons. Let's look at the options. Gain of proton, it is always loss of an electron, not proton. Then question number 232, steroid hormone synthesis, lipid synthesis, chemical detoxification are activities of which of the following? Simple answer to this is, it has to be smooth endoplasmic reticulum. SER is the site of the synthesis of steroid hormones, lipids, and they are actually involved with the detoxification of the chemicals. So the answer to this 232 is smooth endoplasmic reticulum. The next question is, a deficiency of which of the lysosomal enzymes is inherited in a pattern similar to that of Hunter syndrome? Lysosomal enzyme we are talking about. So it has to be Fabry's disease. Fabry's disease, which is actually categorized, which is Fabry's disease, which is X-linked lysosomal storage disorder is due to the deficiency of alpha galactosidase, and it resembles Hunter syndrome where the deficient enzyme is iodouronate sulfatase. What are the other options? Let's look at the other options also. Glucocerebrosidase, Gaucher's disease; sphingomyelinase, Niemann-Pick disease; alpha galactosidase, Fabry's disease; and galactocerebrosidase A, it is Tay-Sachs disease. So the answer to this question is alpha galactosidase leading to Fabry's disease. Next, 234, which of the following cells in the body depends on dynein for its unique functioning? Now here it is lower fallopian tube mucosal cell. The answer to this is fallopian tube mucosal cell. Basically what happens is this dynein is actually essential for the functioning of flagella and cilia. Now out of these, this cilia is actually found in fallopian tube mucosa, by which actually it is responsible to sweep the ovum into the uterus. Dynein is actually found in wherever flagella and cilia are concerned. So the answer to this question is dynein. Next, a small calcium binding protein that modifies the activity of many enzymes and other proteins in response to changes of calcium concentration. Now whenever we talk about small calcium binding protein, the answer is simple: calmodulins. Calmodulins are actually, they require calcium for their function during the process of relaxation and contraction of the muscle. So calmodulins are the basically calcium binding proteins. Then next, baki rest of the options: cyclins, calmodulins, collagen and kinesin. Except calmodulins, nothing can bind with that. Then next question, condition seen in the illustration is due to the deficiency of. यहाँ पे you can see this collar, necklace. This collar, castle necklace is actually due to the deficiency of, this is a characteristic feature of person suffering from niacin deficiency. So the answer is niacin. Now here I would like to tell you some more points about niacin which are actually asked in the exam. Precursor of niacin is tryptophan. 60 milligram of tryptophan are required to synthesize 1 milligram of niacin in the body. Deficiency of niacin leads to pellagra, which is characterized by 3 Ds: diarrhea, dermatitis and dementia, which can lead to 4th D, that is death. So here this castle necklace is the characteristic feature of niacin. So the answer to this is niacin. Then we come over to the last question. True about signaling protein shown in image below. It is simple, the example of interferons. So the answer is host protein. host protein, interferons, which are actually excreted by the host cell in response to the antigens which are attacking our body, like viruses and all. So the answer to this question is host protein interaction. So here we finish up with the discussion of the questions asked in Grand Test 184.